Hello everyone. In today's video, I'll show you how we check the bacterial motility under a microscope. The method that we use is called hanging drop method. This helps us in identifying bacteria that are motile because of presence of one or more flagella. The flagellar arrangement around the bacteria gives them specific types of motility. For the hanging drop method, we require the following: suspension of the bacterial culture that we want to check for motility, depression glass slide, Vaseline, cover slip, nichrome loop, toothpick, and compound light microscope. The first step is to place a cover slip on a clean surface. Next, we apply Vaseline using a toothpick on the four edges of the cover slip. Then we transfer a small drop of bacterial culture suspension using a nichrome loop onto the center of the cover slip. Do not spread the drop. Now take a grease-free depression glass slide and press it inverted onto the cover slip. That is, the concave side should be facing towards the cover slip. Press it gently so that the cover slip gets bound to the glass slide with the help of Vaseline. Immediately turn the slide upwards. You will notice the drop hanging downwards from the slip into the central depression of the slide. Now place the slide on the stage of the compound microscope. The bacteria get accumulated at the edge of the hanging drop because of the surface tension of the drop. That is why we first begin by focusing the edge of the drop under 10x objective lens. Once we can see the edge of the hanging drop, we shift the objective lens to 40x and observe the presence or absence of bacterial motility. The particles in the suspension have a natural motion called Brownian motion. This gives a false impression of motility. So we should be able to differentiate between the true motility versus the Brownian motion. Brownian motion is caused by the water molecules bouncing around in the solution, knocking each other and the microorganisms. In Brownian motion, the bacteria do not change their position much and they are having vibrating motion around a place. Whereas the true motility is when bacteria change their position actively. Now I'll show you some specific types of motility shown by some microorganisms. Dart-like motility is shown by Vibrio bacilli because they have a unipolar single flagellum. It is also known as shooting star motility. Tumbling motility is shown by Listeria species because of the peritrichous arrangement of the flagella on it. Corkscrew motility is seen with spirochetes. They are motile by bending and rotating body movements due to 3 to 4 endoflagella winding around the axis of the cell. Now, if we don't have a depression glass slide, we can still perform the hanging drop method with the help of some plasticine. Rest of the procedure to make the hanging drop remains the same. In this, we make a circle of plasticine and place it in the center of glass slide to create an artificial well. Then we press this slide inverted on the cover slip so that the plasticine binds the cover slip, and after immediately reversing the glass slide, we can observe the bacterial suspension under the microscope. So this was all about the hanging drop method for checking bacterial motility. Do tell in the comment section how you liked the video. Don't forget to like, subscribe and share. Thank you for watching.